you only have 30 minutes to cover the life science syllabus, this is what you should be looking at. DNA code of life, meiosis, genetics and inheritance, and the big rounder of them all, evolution. Evolution counts almost 33% of your paper, followed by genetics, then DNA and meiosis are quite similar in terms of weighting, but meiosis weighs the least. So if you don't have enough time, that already tells about the priority of what topics you should prioritize. You shouldn't be spending more time on meiosis over evolution. Because if you fail evolution, you're not going to do well. Meiosis, on the other hand, you could still get away with it. Let's get into it. So you know the structure for life sciences, right? Let's emphasize this. In my previous paper 1 video, did you watch it? I hope you did. Investigative questions. Drawing a table, not a T-junction. A table is enclosed. There's no space in a table. Graphing can be a pie chart, it can be a histogram, it can be a bar graph. You need to know the kinds of graphs that you could be asked and what they need to have. And the most important is a title. And you need to know if it's a graph that requires an X and a Y component, which one goes here, where does the dependent, where does the independent variable go? What is the aim, what is the hypothesis, and what is the methodology of an investigation? What are fixed variables and controlled variables? Fixed, they are fixed, they do not change. Controlled variables, they could be motivated. Okay, you must know how to calculate percentage increase or decrease. You must know what validity is as well as reliability. Okay, let me tell you, common mistakes learners make for life sciences is not making the answer relevant to the context. And let's talk about this. Improving the reliability of something by increasing the sample size versus you saying increasing the sample size of the plants used. Do you see how now contextualized the answer is? You can't just say increase sample size. Increase sample size of what? The number of teachers? Increase the sample size of your marks? Be very specific, contextualize it to what is being asked in the investigation. Increase the sample size of the plants used. This is very specific to the question. You get that? Graphs are very easy marks. We spoke. Getting the type of graph correct gets you a mark. Remember that histogram doesn't have spaces in between. A bar graph does. The type of graph, getting it, you get a mark. A caption, including both the dependent and independent, you get a mark. The scale lines, the intervals must be the same. So we can't have 0 and then we have 5 and then we have 20. It needs to be consistent and the measurement must be the same, or else you're not going to be able to analyze the overall outcome of what you're supposed to see. Title for each axis, Y and X, you need to have that with units and brackets. Very important. Plotting of points, make sure that you do them accurately. Make sure that if you're given a pie chart, usually you would have your calculations on the side because those would count 2 marks. But I have not seen them ask a pie chart recently. That is also because a pie chart is quite expensive, and I'm going to tell you what I mean by that. To draw a pie chart, you need a certain kind of instrumentation. And that instrumentation, not everyone can afford it. So typically they don't really ask it, but you still need to know it. Okay, let us get into, you need to know the differences between DNA and RNA. Full name, what is DNA and RNA, and you need to know where each of those are found. Okay? One thing I can tell you for free is that you need to know the structure of a DNA. Section A, they'll ask you, is it double helix? What are the building blocks? This is a biological term that's going to come up. Biological term, building blocks, or monomers, it's nucleotides. You need to know what a DNA nucleotide consists of. They can ask you to draw this. The phosphate group, the sugar molecule, and the nitrogenous base. You need to know this thoroughly. There are 4, and they are linked by a type of bond. A what? It's weak. It's a weak what? Hydrogen bond. You need to know the sugar in a DNA and an RNA. If you're asked to tabulate the differences, you must know that. And then make sure that you also practice the pairing. Which one goes with which? For example, we know that in DNA, we know that Adenine goes with thymine, cytosine with guanine. Okay, we got that? One thing I'll tell you for free. DNA replication or protein synthesis is one thing you will always find in a question paper. You will never write a paper without those two. Protein synthesis, they'll ask you, the way they started asking it recently is that they'll ask you to name a process and explain, either transcription or translation. So they'll give you a keyword. Let me tell you. Also know the significance of DNA replication. Where does it take place and when? During interphase in the nucleus. Okay, we're not going to talk about DNA profiling yet. I just want us to focus on this thing quickly. Give me a minute. Let me cook. Give me a minute to cook. Remember that RNA, single-stranded, the other one is DNA, double-stranded. The sugar is different, and then the nitrogenous base is different. We have thymine in DNA, and we have uracil in that one. And then this one has a double helix structure. The other one is a linear structure. Okay? But if you are asked to describe a nucleotide, you need to understand that your answer is going to be separate. Okay. Process of DNA replication, that's fine. Now we need to talk about keywords. If you're asked about DNA replication, remember it takes place during interphase in the nucleus. The double helix unwinds. It has to be a double helix. The hydrogen bonds must be weak. The weak hydrogen bonds break. That's where you get your second mark. The DNA unzips. That's another mark. Each original strand of the DNA acts as a template. That's another mark. Free DNA nucleotides in the nucleoplasm attach to the new complementary DNA bases. Complementary, remember we said A and T, C and G. Complement. Those that did maths, complementary angles. Awesome. So make sure that you follow that. They can ask you to explain various parts of this process, but if you are asked about transcription or translation, you need to understand where each of these happen, because they can say explain a part of protein synthesis that happens in this particular location. And if you don't know what that is, already you are cooked. You need to know what DNA profiling is and what some of the uses. And what are some of the disadvantages? What are the disadvantages of DNA profiling? Remember that it's a very expensive procedure. Got that? You get that. And also when you're given that DNA profiling, you need to check the one that has the most matches as compared. What I used to do is I'd always check, especially most likely they'll ask you a suspect in a criminal case or mother and child for paternity. Mother, you'll always know that this one matches with the mothers, and then see the ones that do not match with the mother but the child has. Who do they match with the most? That is most likely the father, and that is most likely the criminal. Oh, it's awesome. And know what is it used for? For criminals, dead bodies, relatives, and all of that. We're all good. Awesome. Another thing we need to know about this is, remember that translation happens at the ribosome and transcription happens in the nucleus. So that is the tip that you need to know. Very important. Now let us get into the part where people tend to lose marks. This is where a lot of people tend to lose marks, and I think it's about high time we speak about it. Okay, so when we are talking about codons, we have codons and we have anticodons. Remember that there's going to be the codon, the DNA, and then it's transmitted into the mRNA. There's also from the mRNA to the tRNA. So sometimes they'll give you the codon, which is the mRNA. Anticodon is the tRNA. You get that? Remember that when we deal with mRNA, there shouldn't be any T. You should not have T because we do not have thymine in RNA. That is one thing you need to know. Awesome. Awesome. You know the peptide bond, you know the ribosome, you know the organelles and the structures involved. Okay. Another way in which they can ask you the differences, they can say tabulate the two differences between the two monomers. What are the two monomers? DNA and RNA. So you see how getting certain keywords is very important, like a structure, a molecule, an organelle, a monomer. If you don't understand what those keywords are, you're not going to be able to answer. And they give you hints. If we're saying a molecule or a monomer, it's DNA, RNA. If we talk about an organelle, it's a ribosome, it's this, it's that. And very important. And then now when it comes to DNA, the another thing that they like asking you is to choose which protein and whether it would change. Let me tell you how you do that. You first go and see what change that is, and then convert it into your mRNA and then your tRNA where necessary. Usually in the table they'll give you the tRNA sequence, and you need to determine whether if the complementary stand on the DNA changes, is it going to change in the last part. Very important. Once you've seen that, you'll see whether the protein it codes for actually changes. If it doesn't change, then you will say why it doesn't change, because they still code for the same da. Okay. Meiosis, you need to know the differences between meiosis and mitosis. You need to know what a haploid and a diploid cell is. You need to know what a chromosome is. But most importantly here, T keywords: somatic are body cells. Gametes, sex cells. Gonosomes are sex chromosomes. The other chromosomes are called autosomes. And remember, we have 23 pairs of chromosomes. 22 are autosomes. What did we say autosomes are again? All the other chromosomes. Gonosome, gono. Gonorrhea affects the private parts. That's how you're going to remember it. You need to know the different phases. There's meiosis 1 and 2. Similar things happen, but number is a very crucial thing here. Know how to draw the elements. I'm not going to dwell into that one. It's a straightforward topic. Just make sure that you know how to draw. If you're asked to draw anaphase, ana means apart. You need to understand that. Metaphase means middle, equator. Okay, are we there? How do we introduce genetic variation? There are two things in which you can do to introduce genetic variation in the process of meiosis. Crossing over, when does it happen? In prophase. Got that, period. Random arrangement of the chromosomes at the equator. When can this happen? Metaphase 1 and 2. Okay. We spoke about you knowing the differences between mitosis and meiosis. For example, meiosis only happens in reproductive organs. Mitosis happens in somatic cells. But now similarities. Cell division happens in both of them, creates new cells, and both of them start with a parent cell. Awesome. You need to know the process that usually happens when they do that thing. The crossing over, very important. Identifying the various parts of a chromosome, the centromere, the homologous. If you are asked about chromosomes, to label them. Homologous. Homo means same. And then spindle fibers. You need to know what a spindle fiber is. We together? Awesome. Now let's get into the most important topic. Awesome. Let's get into it. Okay. I almost thought we forgot to cover genetics in that other topic, but no, we did not. We spoke about that. We spoke about what happens in the different phases. You need to know that. Okay. Let me tell you what else you need to look at when we do this particular. Okay, let's see. So, random arrangement, remember, metaphase 2. Okay, let us move on to the next. Let us go to this topic, genetics and inheritance. One thing I can tell you for free, they're going to ask you to do a genetic cross. They're going to ask you to do a genetic cross. You need to know certain keywords when we talk about this topic. And let me tell you what are they. Okay, it's not this one. Okay, there we go. We need to know a gene, an allele, a locus, dominant and recessive. If something is dominant, it is always expressed in the phenotype. Recessive can only be expressed if both of those alleles are recessive. Phenotype, pheno, how you look, how something looks, the physical appearance. Genotype is the genetic composition. Homo means same. Hetero means two different. Homophone. Okay, most likely 6 marks. Just by knowing the structure. Remember, you were taught a different way of how to do a genetic cross at school. Follow that one as long as you know that it is in the memo, but I'm sure there's a standard one where people do P1 generation, F1 generation, meiosis, fertilization, and the Punnett square. Please don't make it a T junction, make it a square. And always understand what they're saying. If we're saying homozygous dominant, it's going to be 2 capital whatever. If it is heterodominant, it's 1 small, 1 big. Capital or non-capital. Homozygous, recessive, it's 2 small ones. And you need to understand which one is being expressed. Okay? And make sure you follow that structure because this is something that's going to come up. Okay? Very, very crucial. One thing you need to know here is the recessive, locus, phenotype, gene, allele, monohybrid, dihybrid. Those are the terminologies you're going to find in your section A. Let's finish this topic. We are rushing. You need to know the laws, the independent assessment, and the principles of segregation, but not necessarily usually asked. You need to know complete, codominance, and incomplete. Those 3 you need to know. Section A, they will ask you this always. Okay? Now let us get into the other thing that they can ask you here. Blood groups, they can also ask you to cross those. Very interesting. Another thing I will tell you about this is when they'll ask you about a pedigree diagram for you to be able to analyze trends. And this is usually sex-linked diseases. Hemophilia, for example. You might find also one thing they will ask you is why are males more likely to get hemophilia over women? Men only have 1 X chromosome. And hemophilia is not a trait that is carried on the Y chromosome. It's carried on the X. And that means that a man has a 100% chance. Well, it's 50-50, but with a woman, they can either get it because they have 2 of the X chromosomes. So if that trait is carried on the other one, but not on the other, chances are they might not get it. But with a man, you only have 1. And if you get it, you get it. You get it. So you have a 50% chance. The woman has like 25. The most common, we spoke about hemophilia, color blindness, muscular dystrophy. You don't necessarily need to know the definitions. It would be nice to know for that. Okay, we are done with this particular topic, but that is one thing that they would most likely ask you. Okay, evolution. This topic weighs the most in your exams, and it is very important for you to master it. One thing you know for sure is that speciation is going to be asked, and they might ask you sources of variation that occurs within the same species during meiosis. Point of correction, the random arrangement happens in metaphase 1, not 2. Crossing over, prophase 1. Mutation also happens. Random fertilization and random mating is sources of variation in evolution. Law of use and disuse, you're familiar with that. They don't really. Darwin's theory of evolution, you need to know this one. And contextualize it to whatever is being asked. If they ask you about giraffes that had long necks and short necks, contextualize it to that. Punctuated equilibrium, one thing you need to know is natural selection versus artificial selection. But one thing I can tell you for free, speciation is always there for 6 marks. Make sure you know that. A population of a single species, master speciation. Okay? Natural selection and artificial, we spoke about that. One other thing I want you to focus on here is your keywords. Keywords are very important. If they say natural, it needs to be strictly natural. Don't include anything about artificial. Very important. Awesome. Now, a phylogenetic tree. This is where they're going to ask you questions that are usually level 1, level 2 questions. Characteristics. of the humans versus the African apes. You need to know what is the difference with regards to the foramen magnum. And no, it's not a chocolate. It is not. We need to know what happens to the cranium, the spine, the teeth, the jaw, the palate, the cranial ridges, and the brow ridges between these two individuals. Because you can be asked, and trust that you will be asked, to give differences between these. Humans have a more curved C-shaped spine, whereas with the apes, they have more of a less curved, more C-shaped spine. Okay? And with us, we have more of a curved palate and jaw, whereas with the other ones, it's a bit more like this. Yeah, it's like more rectangular. So please make sure that you understand that. And the hips, who has wider hips and short? Who has narrow and long hips? Of course, it's the chimpanzees. Okay, so this is something I would say you need to go and have a look for. Another thing for this one that you need to go and have a look for is, remember, they can give you a diagram and then they say, tell us why you think this is a human or it's not, it's an ape. Then you need to be able to tell us, okay, the jaw seems more rounded, it's smaller, and there's really not a lot of spaces between the teeth. And there are smaller canine teeth. I'm very passionate about this topic. Why? Because I'm a dentist. Awesome. That is why you need to clock this in. So they might not always ask you to give the characteristics, but they might ask you to say by looking at what is given. For example, if they show you the hip, you can't tell us about the jaw. You see, it don't make sense. It just don't be making sense. You get that, bestie? Awesome. Another thing, we spoke about the 4 reasons of variation. Please know those. Please. If there's anything you want to know those. And then another thing that I want you to know is, why do giraffes have long necks? You need to understand that, because that is what talks about the theory of Darwin. Punctuated equilibrium, know that definition. You will thank me. You will thank me. And also another thing with life sciences, it's not that you fail because you don't know the answer. You just don't represent it in the way they want it. It needs to be contextualized. Remember, I spoke about contextualizing your answer. Very important. Okay. We spoke about the Homo erectus, that one. The differences between humans and African apes you need to know. Opposable thumb, sexual dimorphism, reduced olfactory brain center, you need to know that. And they can also point for you. Give us the differences between this and that of a human and an ape. So you can tell us about the hip because we told you what we want you to tell us. Okay? Evidence supporting the idea that we have a common ancestor. There is fossil evidence, there is genetic, and there's cultural. Very important for you to understand. And another thing is, if you are asked about years, usually it's MYA, million years ago. So make sure that you get that very thoroughly. Lucy, you need to know about Lucy. And the Dawn child and Mrs. Bless. Do you know Mrs. Bless? Yamazu van die Merwe. Gokwam. I'm joking. Let us get into the final aspect of genetics, evolution. One thing that I want you guys to know here is out of Africa hypothesis. Please make sure that you do not forget that thing. Because if you forget that thing, then you might as well not go in right. Okay? Now I just want to have a look and see if there's anything that I might have forgotten. Usually marks for this will go to speciation. Speciation, they'll definitely ask you something there. And then they'll ask you about those things we spoke about, the differences. And then Darwinism, Lamarckism, section A. And then evidence of common ancestral for living hominids. Very important, we just spoke about it. We spoke about the anatomical differences. And you need to also be able to interpret that phylogenetic tree we spoke about. And that is it. You got this. Those are the most crucial things There are other things, of course, but these ones are the ones that are guaranteed to make you pass. Good luck, besties.